Welcome to our video lecture on membrane transport. We are going to build on a lot of the ideas that we gained in our membrane structure lecture and talk about how structure allows for these membranes to do their jobs. Our essential understanding for today is that membranes control the composition of cells by active and passive transport. Plasma membrane is deciding what gets into cells and what stays out of cells, and we do this through active transport and passive transport. Our objectives for this lesson, we are going to distinguish between passive transport and active transport. What do those even mean? We're going to define osmosis and osmolarity. We're going to describe different modes of passive transport and active transport, including endocytosis and exocytosis. And then finally, we're going to talk about dialysis tubing and how we can use it to model membrane transport. Vocab that you should be familiar with from intro bio. If you're not, feel free to pause and look up some of these things. We're going to talk about them in this lecture just really fast. So here we go. First idea is distinguishing between active and passive transport, and this is all dependent upon a concentration gradient. Concentration gradients are simply how much stuff is dissolved. The better scientific word for stuff is solute. So our solute particles are dissolved in our solution. And here we have a high concentration of solute particles, lots and lots of red things in this side of the diagram, whereas over here we have a low concentration, far fewer of those solute particles. We can measure concentration in molarity. Molarity is how many moles of solute dissolved in liters of solution. So here we would have a higher molarity, high concentration, higher molarity. Here we have a lower molarity, lower concentration. So when we are looking at passive transport is going to happen spontaneously. We're moving things from high concentration to low concentration. This happens spontaneously because the universe tends toward chaos, which is energetically more stable. So all these guys being stuck together, this is not stable. There's a lot of energy because these guys are like bouncing off each other. It is more stable to have these guys spread out because there's not as much of that energetic interaction happening because the guys are super squished together. So high energy, unstable configuration at high concentrations. When things are more spread out, they're more stable. There's less of that energetic interactions happening. So again, passive transport, no energy is required because we're moving from high concentration down to low concentration. It's more stable. It happens spontaneously. The universe likes things to be spread out. The universe actually doesn't like or dislike anything. It's just more stable. All due to that concentration gradient. Active transport is the opposite. Now we're going to be moving things from low concentration to high concentration. We're trying to hoard things together. This requires a lot of energy, and that energy is usually in the form of ATP. We're going to look at lots of different kinds of passive transport to get started. So simple diffusion is an example of passive transport. We're moving from high concentration to low concentration across that membrane. And it's just going to happen spontaneously. Things can just pass right through that membrane until we get to equilibrium. Equilibrium is where we have equal concentrations on both sides of the membrane. Things that can simply diffuse across a membrane need to be very small, and they also need to be nonpolar. Stuff that's big, stuff that has a little bit of a charge, things that are polar, cannot get through the membrane because of the nonpolar fatty acid tails. Anything that is polar is going to be repelled by that nonpolar fatty acid tail region, hydrophobic region of the membranes. Things that can pass via simple diffusion, oxygen gas, carbon dioxide gas, and glycerol, which is a little lipid molecule. These guys can all go through membranes via simple diffusion because they are quite small and they're also nonpolar. No charges on these guys. There's no repulsion from those fatty acid tails. We can actually change the rate of diffusion of these molecules. If I raise the temperature, I can speed up the rate of diffusion. Things are just moving faster at higher temperatures. Molecule size, so smaller things move faster than bigger things, simply because they can scoot right through. Something that's big kind of has to ooze its way. And the steepness of the gradient is also going to impact how quickly things can diffuse across a membrane. So, and this is just talking about the differences between the concentrations on the sides of the membrane. So if I have, like, if here's my membrane, and on this side I have 1 molar, remember that molarity is our unit of measurement for concentration. So if I have 1 molar here and 1.2 molar here, this is higher concentration. Things are going to move, but the difference is not very big, so they're not going to move super fast. Whereas if I had like nothing, no solute on this side, oh my gosh, I drew my arrows in the wrong direction. We're moving from 1.2 to 1. We go from high to low. Sorry. If I have 0 molar and like 6 molar, this is much higher concentration. Things are going to move really, really, really fast to where there is nothing until we get to equilibrium. So that's this idea of steepness of the gradient. If we have a big difference, things will move more quickly from high to low. Good. Osmosis. Osmosis is the movement of water across a membrane. And what's kind of quirky about osmosis is we're actually moving from where we have low solute concentration to where we have high solute concentration. And it sounds like this should be active transport if we're moving from low to high, but we're not moving the solute, we're moving the water from where there's low solute concentration to where there's high concentration of solute, but not the water. So the water is moving in relation to the solute. So osmosis is the movement of water across a membrane from low solute concentration to high solute concentration. Here I have low solute concentration, here I have high solute concentration. My semi-permeable membrane, some things can pass and some things cannot. These molecules are too big, they cannot get through the semi-permeable membrane, and so we cannot spread these guys out. Diffusion is not going to happen. And so instead of moving the solutes from high to low, we're going to move the water that's dissolving these solute particles from where we have low solute concentration to where we have high solute concentration. We're going to move, we used to have more water here, some of that water moved over there. And now you can see these guys are spread out kind of equally. So we have changed the molarity of the solute particles. We have equal-ish concentrations just in terms of numbers of particles in space because we moved the water. So the water moves from low concentration to high concentration of the solutes, not of the water's concentration, which is a weird thing to think about because how do we even measure how much water is dissolved in the water? So we don't talk about water concentration, we talk instead about solute concentration. The water is going to move from low solute concentration to high solute concentration. Here's another image showing that, there it goes, where we're moving low solute concentration. So here it's so low it's 0, and we have lots of water molecules. Over here I have high solute concentration, that's those purple guys, and then my water molecules move across that semi-permeable membrane until we get to dilute these molecules a little bit better. Good, good. Osmolarity is simply the measure of concentration of the solute particles in water. So we're looking at solute concentration in water. And we have a couple different pieces of vocabulary to go along with our osmolarity, that solute concentration, where hypertonic, isotonic, hypotonic, hopefully remember this from middle school science or 9th grade bio. So hyper is high, like if you are feeling hyper, you have high energy levels. So hyper is high, we've got a high concentration of solutes. Iso is equal, and then po rhymes with low. Hypotonic is a low solute concentration. So a hypotonic solution where we have low solute outside. So we've got low solute outside, which implies that by comparison, we've got to have high concentration inside. So hypotonic solution outside means that inside the cell is higher concentration. Water from the environment is going to move via osmosis into the cell. And if it's an animal cell, this is really bad because we're going to end up with so much water that this guy could actually explode. The scientific vocabulary for that is to lyse. We're going to lyse that cell. It's going to rupture or burst because it has too much water in it. This is bad, very, very bad for animal cells. But it's amazing for plant cells. So plant cells really like to be in a hypotonic environment. This is their best life. They want to be big and fluffy. They have that cell wall that prevents them from being lysed. So they want to be as full of water as they possibly can be. Isotonic is the happy place for animal cells. We're going to have water moving in and out equally, and so the cell maintains a nice full but not too full volume of water. Our plant cells, they don't really like this isotonic solution. They're going to start to get a little limp. They're going to get flaccid. They're going to lose some of their volume. And the plant is going to start to wilt. Nobody likes a hypertonic solution, so this is where we have lots and lots of stuff, high solute concentration outside the cell, and so water from the cell is actually going to leave the cell and go out. If you eat like a whole bag of chips and you've got a bunch of salt running around your bloodstream, this can actually happen to your brain cells. And so if you eat too much salt and you get a little bit of a headache, it could be that your brain cells are shriveling up like this. Drink more water. Always drink more water. It's good for you. Plant cells really, really don't like this. This is called plasmolysis. The plasma membrane here is actually pulling away from the cell wall. So this plant cell is super, super sad. This happens, so if I have a high solute concentration of fertilizer, if we add too much fertilizer to the plant, we can actually kill it. So don't go overboard with the fertilizer because you do not want to put your plant into a hypertonic environment. Osmolarity, again, this is the concentration of solutes in relationship to cells. And when we have transplants, we need to keep organs, tissues in isotonic solutions because we don't want our cells to dry out. That's desiccation. We also don't want them to lyse. That's when they gain too much water and they blow up. Why? Because we want to keep those tissues alive so that the recipient of the transplant can use them. Because if they're dead, if the tissues are dead, they're not super helpful. And so we've got sodium chloride solution here. Saline solution is going to keep these tissues into a super nice exotonic environment so that the water levels stay super happy inside the cells. All right, we're going to flip away from osmosis. I'm going to talk now about facilitated diffusion, fácil en español, easy. We have some helper proteins that make diffusion easier. So facilitated, we're facilitating, we're helping diffusion happen. This is necessary if I have large molecules or polar molecules like glucose or sucrose. If I have ions that have charges like chloride, calcium, and sodium ions, remember that ions are atoms that have lost or gained electrons. We can have chloride that has negative charges. These are anions. They've gained extra electrons, makes them negatively charged. These positively charged guys have lost electrons. They are cations. If you are in my chemistry class, you know that we talk about how cations are positively charged. Get it? Cat ions. Positively charged. Anyhow, good. So facilitated diffusion is going to be with some helper proteins. We have two kinds of helper proteins. We have channels and carriers. So protein channel is going to be open most of the time, and stuff can just flow through. So again, it might be that this is like a sodium ion. This is non-polar. And so the charge of my sodium ion is going to be repelled by that non-polar fatty acid region, the hydrophobic region of the membrane. And so it just needs a protein helper facilitator to allow it to get through that nonpolar region of the membrane. We're still going from high concentration to low concentration, so it is still diffusion. Now it's just facilitated. We had to add a helper, either in the form of a protein channel or a carrier protein. So carrier proteins, similar to channels, it's just that they're not open all the time. What has to happen is one of our solute molecules needs to bind to the protein, and then it can go through, and then this guy will flip open the other way, something will bind, and that will cause the protein to change shape, and it'll open. So still no energy required. We're still going from high to low concentration, and just the binding of that solute molecule is going to allow the protein to change shape. It does not need ATP to change shape. All right, a super cool example of facilitated diffusion. So this is a gated channel protein, still going to be facilitated diffusion. It is still passive transport, no energy required. But we do have some channels that have gates on them. So we can close the gate or we can open the gate depending on the environment around the cell, the stimulus that is changing the shapes of these proteins. So we're going to talk a little bit in this lecture and then a lot when we get to our unit on physiology about sodium-potassium pumps and sodium and potassium voltage gated channels. So potassium, this is potassium voltage gated channel. So when in nerve cells, we know that they have charges, and if the environment is positively charged, then this gate is going to open. If the cell is negatively charged, then the gate is closed. And so potassium can just flow through that channel when the gate is open, when the channel is open. But if the gate is closed, if the channel is closed, then potassium has nowhere to go. It's stuck either inside or outside the cell. All right, active transport, we're flipping away from passive to active transport. Now we're talking about ATP required. ATP hydrolysis, this is when we take ATP and we turn it into ADP and inorganic phosphate. So ATP, adenosine triphosphate. We have this adenosine thing, and then we have tri is for 3, 1, 2, 3 phosphate groups stuck to that adenosine, which is why it's adenosine 1, 2, 3 triphosphate. What's going to happen in hydrolysis, we're going to chunk off one of those phosphate groups. That's that inorganic phosphate group right there. We now have A with 2 P's. This is a diphosphate, which is ADP, adenosine diphosphate. But what's super cool about this last phosphate group is crazy, crazy energetic. We're going to plug that phosphate group onto one of our proteins, and it's going to transfer that energy from the ATP to the protein so the protein can do its job. That's what's happening here. We have ATP. We're going to transfer that phosphate group to the protein. It's going to give that protein lots of energy, and then that protein can use that energy to move things from low concentration to high concentration. We're going against the concentration gradient this time, against the concentration gradient from low to high. This is not the stable configuration that the universe tends toward, and so energy is required in the form of ATP. We can also use indirect coupling transport. So what's happening here, we're going to move things from high to low concentration. This happens spontaneously, but we can actually use the kinetic energy of that movement to transfer energy to the protein to make something else go from low to high. So this is indirect coupling transport. I can use the energy of the spontaneous high to low movement to make something else move from low to high. So super, super cool. All right, active transport. We talked about channels. We talked about protein channels and carrier proteins for passive transport. Facilitated diffusion. In active transport, now we're going to use the vocabulary word pump. So protein pump implies active transport. ATP is required. If we're talking about passive transport, facilitated diffusion, carriers and channels. Carriers, channels, passive. Pumps are active. ATP is required. So this sodium-potassium pump is implying ATP, active transport. This is how our nerve cells get to the electrochemical environment that they need. And so what's happening is this pump is going to use some ATP to push 1, 2, 3 potassiums out of the cell and pull 1, 2, oh my gosh, did I say potassium? Sodium. I don't know what I just said. Sodium. We're going to move 1, 2, 3 sodiums out. We're going to pull 1, 2 potassiums in. So we're going to move 3 sodiums out, 2 potassiums in. We're going to do that by using ATP over and over and over again. And this is how nerve cells prepare themselves to pass impulses. 3 sodiums get pumped out. ATP is required. 2 potassium ions get pulled into the cell. Again, ATP required. Here's just another graphic, same idea. 3 sodiums are going to bind to the pump. The pump is going to use some ATP to push those potassiums out. Oh my gosh, I did again, to push the sodiums out. Oh my goodness. Sodiums out and then potassiums in. Sodiums out, potassiums in. Good. All right, we're going to flip away from pumps and we're going to look at vesicles. So bulk transport, when I'm moving like big chunks of stuff, so not molecules of glucose or sodium, but now we're looking at huge giant protein molecules or other things that need to get moved around a cell. We're going to do that by making some vesicles. We're going to need both rough ER and smooth ER. The rough ER has those bound ribosomes, so we're going to make some proteins. And the smooth ER is going to make some lipids for us. We always need more membrane in order to make vesicles, but vesicles are formed from membranes. So what's going to happen is we're going to take our, this is the membrane that makes rough ER. Here we have the ribosome bound in the rough ER, the messenger RNA here, and a protein molecule is being formed by the ribosome using the code in the messenger RNA. That protein is going to be transported through the rough ER and then into a vesicle. So the membrane of the rough ER is going to bulge. That protein is going to be in the bulge, and eventually this membrane is going to merge and pinch off. And so now we have our vesicle that can move away from the rough ER. This also happens at the Golgi. So backing up, so here what we just looked at on the previous slide, here we've got our rough ER, we've got our bound ribosomes, we made some proteins. We're going to pinch off that membrane to make a vesicle. That membrane vesicle can then merge with the Golgi apparatus's membrane and dump all that stuff into the Golgi. The Golgi will do its job, and then it's going to make another bulge, which will then get pinched off. That vesicle will move toward the cell membrane, and then this membrane and this membrane will merge, and then all that stuff that was inside the vesicle that was made way back here in the rough ER is going to be secreted outside of the cell. So lots of membranes bulging and pinching off and then merging back into each other. So super cool process. Involved in bulk transport. So again, we're moving lots of stuff all at the same time in vesicles. This can happen because our plasma membrane is held together by very weak hydrophobic associations between the fatty acid tails. So these weak hydrophobic associations allow the membrane to be fluid and flexible. It can bulge, it can move, it can also pinch itself off, and then it can merge with other things. So it is fluid and flexible. It can break and reform, all because of these weak hydrophobic associations amongst the fatty acid tails. Let's talk a little bit about what that, whoops, oh no, I went one too far. Okay, it's okay. Sorry, this recording doesn't allow me to go backwards, and I double clicked. So we're just gonna go. I'm gonna draw a picture of our membrane. So we know that our phosphate heads are hydrophilic. Fatty acid tails are hydrophobic. They are repelled by water. So we've got water outside the cell. We've got water inside the cell. Those fatty acid tails, they are repelled by water, and so they will associate with each other in order to get away from the water. But this association between them, it's super weak, and so these guys can like break apart or stick to something else. They're not bound to each other by real chemical bonds. It's really just a force of attraction holding them near each other, mostly to get away from the water. So we have weak hydrophobic attractions amongst the fatty acid tails, and this is what allows the membrane to do this or that pinchy thing right there. Endocytosis. Endocytosis, we're moving things into the cell. Endocytosis. Endo, in, cyte is cell, moving things into the cell. A couple different ways that we can make this happen. Phagocytosis is when our cells are gobbling up something that's kind of big, like a bacteria perhaps. The membrane is going to bulge and bulge a little bit more and bulge a little bit more, and then eventually it's going to merge over here and then pull that whole bacteria into the cell in a vesicle. Phagocytosis, which is endocytosis for big chunky stuff. We can also do penocytosis. Penocytosis is still endocytosis, but peno is more about drinking. So we're going to make another vesicle, but instead of something big and chunky, we're basically just bringing in particles that are dissolved in water. So we can pinch off a little bubble of water that has some solutes. Again, pinch off that membrane because of the weak hydrophobic attractions amongst our fatty acid tails. And then we can pull these solute particles dissolved in water into the cell. Then we can also use some receptors. We can have some protein receptors that will bind to something, and then the vesicle will form around the receptor and whatever particle it is bound to. This is super important, especially for viruses to try to infect us. So viruses will often take advantage of our receptor molecules to bind to host cells and infect us. All right, we're going to watch a little video of endocytosis. Here goes a white blood cell eating some bacteria. Enjoy the music.
Neutrophils are white blood cells that hunt and kill bacteria. In this spread, a neutrophil is seen in the midst of red blood cells. Staphylococcus aureus bacteria have been added. The small clump of bacteria release a chemoattractant that is sensed by the neutrophil. The neutrophil becomes polarized and starts chasing the bacteria. The bacteria, bounced around by thermal energy, move in a random path, seeming to avoid their predator. Eventually, the neutrophil catches up with the bacteria and engulfs them by phagocytosis.
I'm highly entertained that this super scientific video ends with a burp, a white blood cell burp. Anyhow, good stuff, super good stuff. So that is endocytosis. We're now going to look at exocytosis. So endocytosis was in, exocytosis is out. Endo is in, exo is out of the cell. And so we are going to, we probably had some rough ER back here, and then we had Golgi apparatus here, and we made some vesicles. That vesicle filled with some protein, maybe it's insulin from a pancreas cell or a neurotransmitter from a nerve cell. That vesicle is going to move toward the plasma membrane. They're going to merge their membranes, the membrane of the vesicle, the membrane of the cell, and then the stuff, whatever it is inside the vesicle, can be released outside. This electron micrograph is showing exactly that. We've got some, so here's vesicle membrane, here's plasma membrane, and we can see all the stuff that used to be in the vesicle is being released outside of the cell. Amazing. All right, last little idea for the day. We are almost done, my friends. Dialysis tubing can be used to model membrane transport. So dialysis membranes are semi-permeable, just like plasma, well, not just like plasma membranes, in some ways similar to cell membranes. And so plasma membranes do more than dialysis membranes, but dialysis membranes similar in that they are semi-permeable. So here we are going to start in our dialysis tubes with some stuff. So we've got some big solute particles and some little solute particles. Perhaps this is like a starch molecule, which is quite large. Perhaps, starch. And perhaps this is sugar, which is a little bit smaller, glucose perhaps. And over time, what's going to happen is that the small particles are able to get out of the plasma, the dialysis membrane, because there are some tiny, tiny holes, and so tiny, tiny things can get out. And so we will have glucose outside of the dialysis membrane after a while. However, the starch molecules, or whatever this big guy is too big, it cannot get out. So at the end of the investigation, we will still have those large molecules stuck inside the tube. What has to happen to deal with those big things? Well, we're probably going to also have some water moving in to the dialysis tube in order to dilute those big particles that cannot get out to get to equilibrium. Good, good. All right, let's quiz ourselves just a little bit. If I have distilled water here and I have some starch outside my dialysis tube, do you think that the solution inside the tube is going to test positive for starch? Can the starch get into the tube? The answer is no. Starch is too big. We are not going to have any starch inside the tube. What's going to happen, though, is our tube is going to lose mass because water is going to leave the dialysis tube and go out to dilute the starch that cannot get into the dialysis tube. All right, which environment is hypertonic and which is hypotonic? Remember that hyper is high, 2 is higher than, well, if it's just water, it's 0. So our 2 molar starch solution, this is hypertonic and this is hypotonic. High on the outside, hypo rhymes with low, less starch on the inside. All right, one more little question. Is the dialysis tubing going to gain or lose mass? Can the starch get in? It cannot. So what's going to happen is that water is going to exit in order to dilute the starch that is stuck outside the dialysis tube. The dialysis tubing is going to lose mass because water left. And that is our day, my friends. Just a double check, make sure that we accomplished our objectives. We did distinguish between passive transport and active transport. We talked about osmosis, movement of water across the membrane from high solute concentration to low solute concentration. Osmolarity is just that concentration of solutions around our cells, hypertonic, hypotonic, isotonic. We talked about different ways of passive transport and active transport. Passive transport, simple diffusion, facilitated diffusion. Active transport is going to be using ATP with pumps, or our bulk transport definitely needs lots and lots of ATP. We did define endocytosis, exocytosis. Endo is in, exo is out. These are both forms of bulk transport. We are building membranes and moving things around. This is definitely going to require some ATP. And then we talked a little bit about dialysis tubing, and that, my friends, is our day well done.